so let's cover the muscular system, give you a quick overview. So, muscle cells are muscle fibers, okay? That's kind of the bottom line. We use those terms interchangeably. Microfilaments, when those move, muscles contract and shorten. Muscles are going to share some terminology. So if you see things like myo or mys, it means muscle. And also, if you see sarco, it means muscle or flesh. Okay? And again, we're just going to hit the highlights here, so we're probably not going to spend a whole lot of time on each slide. I'm pulling it straight from Canvas, so if you miss it, just go back and pull it up. All right. So 3 types of muscle: skeletal muscle, cardiac muscle, smooth muscle, right? 3 types, okay? Y'all good there. So just give you a quick overview. Skeletal muscles attached to the bones. Cardiac muscles attached to, is in the walls of the heart. Smooth muscles in the walls of hollow organs. All right. Skeletal muscles, these long rows here. It has these lines of striations. Cardiac has striations, but it's branched. It's not straight lines across. Smooth muscle has no striations, and the cells are narrow. All right, so they are kind of, we call them spindle shaped. They're pointy on the ends. All right, so again, smooth muscle, no striations. Cells are spindle shaped like this. 1 nuclei per cell. They're involuntary, so you don't have to think to make their food go through your intestines, all right? So again, you find them in the walls of hollow organs like your stomach and urinary bladder. All right, so shows you here's some layers of smooth muscle. So this is a circular layer. Okay, you can see the spindle fibers, and then this layer around it is more longitudinal, so you see it in cross section. Okay, but it's still all smooth muscle. It's just the way it's cut. All right, cardiac muscle, again, found only in the heart. It's involuntary. You don't have to think to make your heart beat, right? So each muscle cell or muscle fiber is connected to each other by what we call intercalated discs. Those are these little dark lines. These are branched, so they're not rows straight across. 1 nucleus per cell. Okay? So they have that in common with smooth muscle. And they are striated. You can see little lines in there. Skeletal muscles, okay, they're attached to the bones. Multiple nuclei per cell. That's what all these little things are in here. These are nuclei. That makes them unique. Skeletal muscle unique. They have these lines, visible bands of striations, but they are voluntary. So that's unique to skeletal muscle as well. You do have to think to make your muscles move. Okay, functions of skeletal muscle. Allows you to move, right? Helps you maintain your posture. So you may not even be, you're not thinking about it right now, but how you sit, how you stand, how you walk, your muscles are helping you maintain your posture, okay? So muscles help you stabilize your joints, okay? So keep your joints in good shape. And they generate heat when they move, okay? They generate heat. Okay, connective tissue. These are wrappings of skeletal muscle, right? So you'll see these, see connective tissue wrapping the skeletal muscle. So we have different layers. So endo means inside, right? Or inner. So it encloses a single muscle fiber. It's a layer of connective tissue. Then perimysium will wrap around a bundle or fascicle of muscle fibers. And then the epimysium will cover your entire skeletal muscle. So endo, peri, epi. Okay? You also have fascia, which is outside your epimysium. Okay, so it shows you what this looks like. Here's one individual muscle fiber cell. Okay, in this little space in here, around each one, you have endomysium. So that's the space in between all of these muscle fibers. Not to be confused with this wrapping that goes all the way around it. The wrapping that goes all the way around it is perimysium. Okay, forms a fascicle. All right, now around your whole muscle, you have epimysium. Okay. So endomysium in here, perimysium around, and then epimysium all the way around. All right, so the epimysium is going to basically blend in with the connective tissue that attaches the muscle to the bones, the tendons. Okay, and they're made up of collagen. All right, another kind of connective tissue you'll see also are aponeuroses. These are sheets of connective tissue. And they can attach bones or attach muscles to bones or cartilage or other connective tissues. Okay. Just like, we talked about cells, remember the plasma membrane, right? So we give it a special name. Remember we said sarco has to do with flesh or muscle? So we call, and lemma means plasma membrane. So basically, sarcolemma is a muscle cell's plasma membrane. All right, now inside the muscle cell, you have these organelles. Remember, those are those different components of the cell called myofibrils. Got light bands and dark bands in them. Okay, and that's where you see the striations. So abbreviation for light bands, the I in light, get I bands, second letter, right? Second letter. Second letter of dark is A. So these are A bands, A and I, okay? All right, so this is one muscle fiber. Now, again, if you go higher magnification inside, you can see all these myofibers. Okay? Here you can see, here's a dark band or an A band, light band, I band. Okay, and all these nuclei. And you need all those nuclei because remember, nuclei have what inside of them? You gotta remember what's in the nucleus? DNA. What do we use DNA to make? Indirectly, yes, but what's that process? There's two processes. So DNA is used to make RNA in transcription. That RNA leaves the nucleus and makes protein in translation. So you need lots of nuclei so you can make all the proteins for these muscles. All right. So banding pattern. Again, light band, dark band. Okay, light band has thin filaments in it. Thin filaments only. Dark bands are the length of the thick filaments. So there are going to be some thin filaments in them, but it goes the whole length of the thick filaments. All right, and we'll get into these others. I think will make more sense when we show you the picture. All right. So this is a myofibril. All right, say hi, myofibril. Okay. All right, thank y'all for playing. Okay, so you can see the thicker lines. We call those the thick filaments, made up of myosin. The thinner lines, the blue lines, we call those thin filaments, and they are made up of actin. All right, myosin is a thicker word, thick myosin, thin actin. All right, so y'all see how this line kind of zigzags? So zigzag starts with the letter Z. So this is your Z disc, okay? All right. Now, this line here, what I want you to do is to be kind of creative. Kind of turn your head like this a little bit. You see this line here, and that line there. If you turn your head, it looks like a letter of the alphabet. What letter does that look like if you do?
H.
H. So this is the H zone. Okay. All right. So you got your H zone and your Z disc. Your H zone right here is in the middle of your thick filaments, right? In the middle of that H zone is the third. You see the little third line in there in the middle? It's in the middle, so we call that the M line. Okay? All right. Z disc is in the middle of the thin filaments. Does that make sense? Do you see that? H zone and the M line in the middle of the thick filament. So what's going to happen here is when muscles contract, the Z discs get closer together. Okay? That makes a contraction. The thick filaments don't really move. It's the thin filaments that move when the muscles contract or relax. All right. So what we call a contractile unit, so if you look at each individual unit that's contracting, we call that a sarcomere. Remember, sarco means muscle or flesh. So what's in it? Thick filaments and thin filaments, myosin and actin. And the alternation creates the stripes or striations. All right. Thick filaments, again, myosin. It's myosin protein. Okay, has enzymes in it. Remember enzymes? There's a class of protein, right? And this one is has ATPase enzymes, which means they break down ATP. Okay. And they have what we call myosin heads. So these look kind of like little golf clubs, right? So the heads we call these cross bridges when they link up with the thin filament. All right, the thin filaments are the actin filaments that have the actin protein. And the Z discs are the anchors for your thin filaments, right? They're in the middle. So when you're at rest, there's a zone that's where there are no thin filaments, no actin. That's your H zone. All right, remember we showed you the H zone? All right. Remember, on either side of the M line forms your H zone. You see how there's no thin filaments right here? Just thick filaments? Okay? That's your H zone. So there are no thin filaments. And a sarcomere is basically Z disc to Z disc. That's the distance of your sarcomere. All right. So anatomists, we love these, long names, right? So in the muscle cells or muscle fibers, we have sarcoplasmic reticulum. Where you get all been out of shape about that. All it is smooth ER. If it's smooth ER in a muscle cell, has some specialization to it, it's a sarcoplasmic reticulum. Okay? Everybody say sarcoplasmic reticulum.
Sarcoplasmic reticulum.
All right, very good. So y'all can impress everybody at home when you go home today. You can say sarcoplasmic reticulum. So what does it do? It stores calcium and releases it. All right. So in calcium, you think about it for your bones, but you need it for your muscles too. Okay? And it surrounds your muscles or myofibrils in your muscle cells. All right. So we're going to transition here to characteristics of skeletal muscle cells. One is irritability. No, it doesn't mean they get upset. It means responsiveness. Okay? So this means they can receive a stimulus and respond to it. Contractility means they can shorten, they can contract when you get the stimulus. They're extensible, which means they can be stretched. So it's kind of the opposite of contractility. Okay? If you think about it another way, extensibility is the ability to extend, right? Elasticity means recoil and resume resting length. So you can stretch it, and after you stretched it, it goes back. Okay, anybody ever bought a slinky? You got a real slinky, you can play with it for a while, but you buy a slinky from the dollar store, you stretch that thing a couple of times, that thing don't work anymore, right? So it is not very elastic. You laugh, but y'all understand, right? So, all right. So nerve stimulus, action potential. All right, so skeletal muscles, they gotta be stimulated in order to get a contraction, all right? So a nerve cell or neuron has to stimulate the muscle cell. Now, when you have a motor neuron, it causes an action in the muscle, and any skeletal muscle cells that are stimulated by that neuron, we call that a motor unit. Okay, some neurons stimulate one muscle fiber, some stimulate multiple muscle fibers. But they're all considered a motor unit because they're related to that one neuron. All right, so shows you an example here. So here's motor unit 1. You have your nerve. All right, and you have this muscle cell and this muscle cell being innervated by it. So that's one motor unit because they're being innervated by the same neuron, same nerve cell. Motor unit 2 has 1, 2, 3 muscle fibers that it innervates, right? All right. Shows you you're on a microscope, same thing. All right, so these are different muscle fibers being innervated by nerve cells. All right. Neuromuscular junction. So what's a junction? It's a place where things come together, connected, right? So neuromuscular, what's neuro? Nerve, what's muscular? So it's a place where muscles and nerves connect. So fancy way of saying it is it's the association site of axon terminal of the motor neuron and sarcolemma of muscle, right? And what's the sarcolemma? The plasma membrane. Okay, nerve cells touching the plasma membrane. Okay, neurotransmitter. So this is a chemical that the nerve releases, okay, when the nerve signal goes all the way through the nerve, gets to the end of the terminal of the axon on the nerve. So for skeletal muscle, the neurotransmitter is acetylcholine, okay, or ACH. You'll see those two terms. That's a neurotransmitter. All right, so what's transmit mean? Send, right? So it's a chemical that a nerve sends a signal through. That's all it is. All right. Now, a specific area in the neuromuscular junction we call it synaptic cleft. So it's the gap between the nerve and the muscle, and they're not making contact there. So if you got to think about this from a microscopic standpoint, it's so small that it's that little bitty space in between that nerve, into that nerve and the muscle cell. Okay? It's filled with interstitial or extracellular fluid, right, in the space in between. All right. So transmitting that nerve impulse to muscle. So the nerve impulse is reaching your axon potential, or your axon terminal. So nerve signal goes all the way to the end of the axon of the nerve. That's what that says. So then on that on that muscle cell, channels for calcium are going to open up. And then calcium ions, actually, I should make this more clear. These channels are on the nerve. So like on the edge of the nerve, there's openings for calcium. Those calcium channels open, calcium goes out. The calcium ions are going to essentially trigger synaptic vesicles, which, anybody remember what a vesicle is? Those circles? Anybody remember seeing those little circles? The calcium makes those vesicles hit the edge of the axon of the nerve. And they open up and let the acetylcholine out, let the neurotransmitter out. All right, and then they're gonna, the acetylcholine then diffuses, remember diffusion, moves from area of high concentration to low concentration? So it just goes across the synaptic cleft because there's less of it over there on the other side. It attaches to receptors on the plasma membrane or sarcolemma of the muscle cell. All right, so it's basically this whole process is basically a way to move acetylcholine across the synaptic cleft. Okay? So that's why I made you learn about diffusion. Okay? Because this is one of the ways where you see it. All right. You get enough acetylcholine release, the plasma membrane of the muscle cell becomes permeable to sodium ions. Remember, permeable means things can pass through, right? Go across. Sodium's going to go in the cell then. Okay? And potassium is going to come out of the cell, of the muscle cell. And this leads to what we call depolarization. Makes more sodium channels open. And even more sodium goes in the cell. Okay, when you get to a point where there's depolarization, it basically does something what we call all or none. And so once you start and you get an action potential, you can't stop it. Okay? And then you get a muscle contraction. Okay, so either you have enough charge difference to create a contraction or you don't. Okay, now, what if this went on forever? Your muscle, you couldn't stop your muscles, right? Like you'd just be stuck, like you'd be having a cramp, right? You have to have a way to stop this. So acetylcholinesterase, or ACHE, will break down your acetylcholine. Okay, it breaks it down into two things, acetic acid and choline. So it's decomposition, remember that? ACHE is going to end the muscle contraction because it's moving that ACH so it can't make that muscle contraction keep happening. So how do you get back to resting state? Well, basically if potassium moves out of the cell, you can get back to resting state. You also have the sodium-potassium pump. Remember we learned about active transport? It's going to move sodium and potassium ions back to where they were originally. Okay, so those are ways you get back to resting state. This is a visual. Here's your neuromuscular junction where the neuron and the muscle touch. Here's your synaptic cleft, the little space in between. All right, action potential or reaches what we call the terminal of the axon, the ending. All right, so it's causing calcium channels to open. Calcium rushes in to the neuron, attaches to these synaptic vesicles. They go to the edge and release their acetylcholine. Acetylcholine, and these receptors here on the other side say red rover, acetylcholine come over. And the acetylcholine comes over and attaches onto these receptors because of diffusion, right? Because there's less of it over here. And then you get trigger of action potential on the muscle cell. You get a contraction. All right. And again, there's that vesicle releasing its contents through exocytosis. Remember, exo means going out. Acetylcholine breaks down the acetylcholinesterase breaks down the acetylcholine so the contraction can stop and close the channels. All right. Gives you an analogy comparing your action potential to a flame. Right? If you burn this piece of the wood, it's going to spread, right? Same here. If you have an action potential here, it's going to spread along the muscle fiber. Okay. All right. So let's pause here. Talk to your partner about 3 things that you have learned so far about muscular system. 3 things you've learned so far. And I don't know is not an acceptable response. Yes, ma'am.
Christian flexible.
Thank you for letting me know. Okay. All right, so let's keep going here. All right, so mechanism of muscle contraction. And calcium is going to bind to regulatory protein, right? On those thin filaments, the actin. Calcium binds to actin. Exposes sites on actin that are for binding to myosin. So it allows actin and myosin to bind to each other. When that happens, the myosin heads do what's called a pivot. And it slides the actin, the thin filaments, toward the center of the sarcomere. It allows the sarcomere to shorten. You get a contraction. And this happens because of ATP. You use energy to do it. So you need ATP and you need calcium ions. All right. This shows you relaxed sarcomere where you have a large 8 zone. And when you have fully contracted sarcomere, you have a very short, or basically almost non-existent 8 zone. All right. And again, remember this is the I band where you only have thin filaments, and the A band where you have the length of your thick filament. So again, when actin is in a relaxed state, actin, the sites on actin that bind to myosin are basically blocked. Okay, so when you get calcium ions there, those sites get opened up, and there it's no way actin and myosin can bind together and move the actin towards the center of the sarcomere. All right, so that's what's happening here. You get calcium ions, and then here you start getting a pivot. So look at, you see where the myosin heads are here, and then you see here how they move downward. Okay, so they're pulling them toward they're pulling the thin filaments toward the middle. All right. So that brings us to contraction of skeletal muscle. As I said before, it's all or none. Either you contract or you don't. So within skeletal muscle, only some fibers might be stimulated. You might not stimulate all the fibers. And different combinations of your muscle fiber contractions will give you different responses. Okay, so you get what we call graded responses, which means you have different levels or degrees of the shortening of the skeletal muscle. All right, so you have graded responses, and these are a factor, or these are influenced by how often the frequency of the muscle stimulation, or the muscles being stimulated. So if you have very rapid stimulation, you have a larger response, right? If you have very infrequent stimulations, you have a lower response. And the number of muscle cells that get stimulated. Okay? So if you have a lot of stimulation, but only a few cells, muscle cells stimulated, not as much as if you had lots of stimulation and lots of fibers being stimulated. So I guess one way to think about that is if I have on my desk, if I had like 5 buttons, right? So if I push one button every so often, that just gives you a little bit of stimulation. But if I have 5 buttons and I'm pushing them very fast all the time, more stimulation, right? Higher response. Okay. Twitches are just brief and single contractions. And these actually technically are not normal to get muscle twitches. All right, shows you that when you get that brief contraction, you get an increase in tension. All right, because when you're contracting, you're generating some tension. Now, contractions can also be summed when you have multiple contractions. So if one contraction stimulated, you followed up by another contraction, you get more stimulation than the individual. So it's kind of a synergy. So it's kind of like 1 + 1 is 2, but if 1 and 1 are working together, you can create some reaction that's more than 2, kind of thing. So, and this happens because the muscle doesn't get to completely relax. Right? So if you get some tension and before you go all the way back down to no tension, you stimulate again, you're going to go higher than you would by just one contraction itself. And that's what you see here. Muscle fiber didn't get to completely relax, so you got higher tension. So if you had waited, you'd have had basically 2 equal curves. Does that make sense? So because you didn't wait, you actually got higher tension. All right. Term that you'll see is tetanus, and here we're not talking about tetanus when you step on a rusty nail. This is actually, we're talking about it in the context of muscles. So it's called unfused or incomplete tetanus. So here you get some relaxation occurring between the contractions with the nerve stimuli or arriving at a faster rate because of this summing. Or actually, it's even faster than what you get during summing and contractions. And unless this muscle contraction is said to be smooth and sustained, it's said to be in unfused tetanus. All right. So here it being unfused, you have continually having more stimulation and not allowing the muscle fibers to relax, right? Can y'all see that the muscle fiber didn't get to go all the way back down to 0 in tension? And so you kept getting increased tension. So we call this unfused tetanus because it's not continuous. It kind of goes peaks and then goes back down, peaks goes back down. But the overall direction is up. Whereas with fused or complete tetanus, you get no relaxation at all. All right, so the frequency of your stimulations is not allowing for relaxation between the contractions. The result gets you smooth and sustained muscle contraction. These stimulations are so fast, you get smooth lines for your tension. Okay? So obviously there's a point at which it's as high as you can go. It slows down at some point with the rate, but it's a smooth line instead of all the little peaks. Okay, so the muscle and how it responds to strong stimuli. So again, just like tension is affected by how many fibers are stimulated, the overall force that muscle puts out is going to depend on the number of fibers. So contraction here of more fibers is going to result in more tension. And the muscles are going to be able to continue to contract unless you run out of an energy source, right? So as long as you have the conditions, you're going to get contraction. So where does that energy come from? Well, I'm glad you asked. It's ATP. All right, you learned about that already. So that's your immediate source of energy to get your muscles to contract. It's stored up in muscle fibers in small amounts. You use them up. And then once you have used up your ATP, you got to find other ways to make it, right, when you run out. So how do we make more ATP? We have direct phosphorylation of ADP by what we call creatine phosphate. And before you begin, like, well, what's phosphorylation? Well, let's look at the beginning of it. What's this? Phospho, which means phosphate. ADP has how many phosphates on it? What's the D stand for? You got to remember? Di. Di. So Di means what number? 2. The T in ATP stands for 3. So where's the phosphate coming from? Just told you. Creatine phosphate. So it's essentially a substitution reaction, right? So you're just moving this phosphate over onto ADP to create ATP. Also, we have what we call aerobic respiration. So again, we're not really talking about aerobics like doing jazzercise. What we mean is aerobic means uses oxygen, right? So if you're doing exercise, you're probably going to be needing some oxygen. So it's aerobic. All right, so it's using oxygen to make ATP. And then you have anaerobic glycolysis. And anaerobic means without oxygen. All right, so these two processes, glycolysis and lactic acid formation, make you a small amount of ATP compared to aerobic respiration. So when they talk about feeling the burn, it's this acid in your muscles. Okay, when it accumulates and feels like your muscles are burning, that's lactic acid buildup. So it gives you some, but you need to be able to generate more ATP usually. All right. So it's direct phosphorylation by creatine phosphate. It's the fastest way of making it, because all you got to do is, disattach it from creatine and put it on ADP. Your muscle cells will store creatine phosphate, so you can use it that way. All right, and then once you deplete your ADP, you get ADP. Creatine phosphate is going to transfer its phosphate over to ADP, create ATP. But your supplies of creatine phosphate usually get exhausted in less than 15 seconds. All right, so it's not going to sustain you very long, right? And generally, per molecule of creatine phosphate, you can only generate 1 molecule of ATP. And you need a whole lot more than that. Because again, you're just substituting, all right? So this is a reaction here, creatine phosphate and ADP gives up its phosphate, you get creatine and ATP. You don't need oxygen, you get 1 ATP per molecule of creatine phosphate, and only lasts for 15 seconds. Aerobic respiration, your glucose gets broken down into carbon dioxide and water. And you get 32 molecules of ATP, right? So per molecule of glucose, you get a whole lot more ATP from aerobic respiration than creatine phosphate, right? So you get a whole series of what we call metabolic pathways in your mitochondria. Metabolic just has to do chemical reactions, okay? And this is slower, right, because it takes more steps. And it requires continuous supply of oxygen. And as byproducts, you're going to generate carbon dioxide and water. All right, so, and you think about that, why, ever thought about why you have to exhale carbon dioxide? We talk about breathing in oxygen, exhale carbon dioxide. Because your muscles are generating it, and you got to get it out. Okay? All right. So where do we get this ATP from? The glucose, sugar. All right. Break it down to pyruvic acid. Pyruvic acid goes to your mitochondria, makes ATP, 32 of them. And water and carbon dioxide. So this takes hours. Okay? So if you eat something, you're probably not going to generate lots of ATP really, really quickly just right after eating. It's probably going to take a little while to break it down. All right, anaerobic glycolysis, lactic acid formation. So here you have reactions, breaking down your glucose, but it does not need oxygen. Okay, it does not need it. When you break down your glucose, it breaks it down into pyruvic acid, makes 2 ATP molecules. And if you don't have any oxygen, your pyruvic acid is going to be converted to lactic acid. And it's not efficient, but it is fast because there's not that many steps involved. So this reaction's not as efficient, but again, you need huge amounts of glucose because you can only generate 2 ATPs. So in order to generate more ATP, you gotta have a lot more sugar. Lactic acid is gonna produce muscle fatigue. Right, so that's this part here. Glucose breaking down, making pyruvic acid, generating lactic acid. All right, 2 ATPs. May take 40 seconds to do so, but you don't need oxygen. All right, so if you get strenuous and prolonged exercise, you're going to get muscle fatigue, right? You get tired, your muscles get tired. You get what we call ionic imbalances. You get a lot of lactic acid in your muscles, and your supply of ATP decreases. So once you finish exercising, we have what we call an oxygen deficit. And how do you repay that? Breathing deeply. Okay, you're not just breathing deeply because you're tired, you're breathing deeply because that's your body's normal reaction to get more oxygen and generate more ATP, right? All right, contractions. So one type of contraction is isotonic. All right, the myofilaments are going to be able to slide past each other during the contractions. Muscle shortens and you get movement. Like when you bend your knee, you rotate your arms. So most of the time, when you're having, these are not what you normally see, or isotonic contraction. And remember, iso means same. So in isometric reactions, you're actually increasing the tension, right, but the length isn't changing. All right, so the muscle is unable to shorten or produce movement. So it's again like if I'm pushing against the wall, there's no change in movement. I'm contracting, I'm pushing, and I feel the contraction in my arm, but there's no movement. Okay, so there's not necessarily going to be a change in length of the muscle. Okay. Muscle tone is going to keep your muscles healthy and ready to react. It's a result of a staggered series of nerve impulses that are delivered to different cells in your muscle. That gives you muscle tone. And if your nerve supply gets damaged, your muscle is going to lose muscle tone. And it can become paralyzed, and it can also atrophy. Atrophy means decrease. All right, so you need a certain amount of muscle movement to keep your muscles moving and living normally. Like astronauts, and they're in a weightless environment, they don't push it off much, they're not using their muscles much. Their muscles atrophy, and they come home they're extremely weak. So how does exercise affect your muscles? Well, it can increase the size of your muscles, the strength of your muscles, and the endurance of your muscles. So aerobic endurance exercise, so aerobic has to do endurance. Things like jogging, biking, gives you stronger and more flexible muscles. And they're more resistant to fatigue, so you don't get as tired. Also makes your body's metabolism more efficient. Okay, so it helps your body do its chemical reactions better. Improves your digestion and your coordination. Now, resistance or isometric exercise like weightlifting is going to increase the size and the strength of your muscles, right? So both have value, but they do different things, right? So aerobic is more for endurance, isometric is more for size and strength. All right, so you can see the difference there. So you can look at the difference between this person's muscles and that person's muscles, right? Because this person is doing endurance, aerobic exercise, this person does isometric exercise, right? And then the body adjusts. All right, we'll finish here with 5 golden rules of skeletal muscle activity. So, generally speaking, skeletal muscles cross at least one joint. The bulk of your skeletal muscle is going to lie proximal or near the joint that's crossed. Skeletal muscles typically have at least, or all have at least 2 attachments, an origin and insertion, and all that means is they got to connect in 2 spots, right? Because if they don't, it's hard to move against it. Like your biceps brachii is going to have an attachment at the top of your arm and the bottom of your arm, right? So, because it's got to move 2 things, right? Usually it's going to be moving two bones. Skeletal muscles are only going to pull, they don't push, right? So, they're pulling because they're shortening, right? So, even when you're extending your arm, what you're really doing is contracting your triceps brachii, right? And during contraction, your skeletal muscle insertion is going to move toward the origin. So again, like the biceps brachii example, this is my origin, this is my insertion. When I contract, I'm moving my insertion closer to my origin, okay? And actually my insertion is down here, so it's getting closer, right? So that's the muscular system. So let's actually finish up by doing your exit tickets. All right. Giving your clear points and your muddy points.